Of course, those aren't the only cells of the innate immune system. We do have other cells that are classified as innate. You may remember in the previous set of slides when I showed you that diagram of all of the different cells that can be differentiated from a hematopoietic stem cell. I pointed out one type of cell that comes from the lymphoid lineage that actually is an innate cell. So they resemble CD4 positive cells. They produce similar cytokines, but instead of responding to particular antigens like a T cell would do, they respond to those pathogen-associated molecular patterns and damage-associated molecular patterns. And those cells are called natural killer cells. Natural killer cells are great because they do give a general response to viral infection. They will induce virally infected cells to undergo apoptosis. They have anti-tumor activity. They can increase the inflammatory response, which, again, the inflammatory response is not a bad thing. Having a fever is not a bad thing. It's when you have too much that becomes a bad thing. And they also mediate what's called antibody-dependent cellular cytotoxicity. So if a cell is coated with antibodies, they can cause the destruction of that cell. Other cells of the innate immune system include natural killer T cells and gamma delta T cells. And it's just the slide formatting. It should be lowercase gamma delta. I don't know why it's capitalized gamma delta. I couldn't make it go lowercase, but that's okay. They're considered to be cells of the innate immune system, even though they are a type of T cell, because they have very limited T cell receptor diversity, meaning they don't respond to specific antigens like a regular T cell would. They do recognize specific types of antigens, for example, bacterial phospholipids or various bacterial metabolites. And one of the major things that these things do is they produce interferon gamma. And interferon gamma is a super, super potent pro-inflammatory cytokine. And that can activate macrophages and dendritic cells, which are super great phagocytic cells, to come in and help clear out an infection. Here, what we're trying to show you is various infectious agents and how those infectious agents are recognized by the innate immune response. So viral glycoproteins are recognized by toll-like receptors on the cell surface. Bacterial lipoproteins, bacterial peptidoglycan, lipoteichoic acid that's found in gram-positive cells, the LPS that's found in gram-negative bacteria, flagellar proteins, various carbohydrates and cell wall components of fungi, and these anchored proteins that are components of protozoa are all recognized by a couple of different, mostly toll-like receptors found on the surface of the cell. Now, with viruses, their main goal is to get inside the cell, and when they do, the viral nucleic acids can be recognized by other toll-like receptors and other receptors of the immune system. If bacteria get internalized, either through phagocytosis or through invasion, their DNA or peptidoglycan can also be recognized by receptors. And then if some of the protozoans get in, things like malaria are intracellular, or if they're phagocytized and their DNA gets in, again, you have that toll-like receptor response. So you have receptors on the inside and on the outside of cells that can stimulate the immune response. And so these things here, these glycoproteins, the DNA and RNA, the lipoproteins, the peptidoglycans, the LPS, flagellar proteins, fungal carbohydrate, these are all considered to be PAMPs. They're pathogen-associated molecular patterns. When you have this type of recognition, so when you have infection, so here they're showing you the lipoteichoic acid, the LPS, the flagellar proteins, the internal DNA or RNA, you get a lot of changes in gene expression in that cell. And you can have activation of the innate response, more antimicrobial peptides, more inflammatory cytokines, interferon production. And then you can also drive the adaptive response. You produce cytokines that recruit helper T cells, T helper 1, T helper 17. The other thing you can get, though, with tissue trauma or infection, even like exposure to toxic chemicals like silica or asbestos, you get this structure that forms called the inflammasome. And the inflammasome leads to the production of more pro-inflammatory cytokines and just drives a generalized inflammatory response. So it's a cycle here that is, at the end of the day, leading to inflammation. Here we're showing what chemotaxis looks like. So you have the release of cytokines, these chemokines, right? Specific type of cytokines that recruit various cells of the immune system. So here they're showing a neutrophil traveling through the blood vessel, and it will eventually leave the blood vessel and try to get to the tissue. And they're showing it here again. You have a neutrophil, and the steps to this rolling, right? The cell will come into contact with, so the immune cell will come into contact with the endothelial cells lining the blood vessel. So rolling is kind of a generalized contact as it gets closer and closer to where these chemokines are coming from. The cell will become activated and more tightly bind, even more tightly bind. And then you have this step here called diapedesis or extravasation, where the immune cell comes through the endothelial barrier into the tissue. This action of the diapedesis or the extravasation, it's called either or, is what leads to fluids and red blood cells that can also enter the tissue, causing the swelling and redness that we see. Phagocytosis, again, another one of those generalized innate immune responses. So here they're showing there's a couple of different ways. Bacteria can be recognized if they're coated with antibodies. The cells of the immune system, like macrophages, can recognize the FC region, the constant region of an antibody, but they can also recognize, using those toll-like receptors, some of the PAMPs associated with the bacterium. So they're internalized, and most often these antimicrobial compounds, including things like reactive oxygen species, will lead to the destruction of the microorganism. What we'll talk about with, unfortunately, a great number of pathogens is that plenty of them are able to be phagocytized but avoid destruction. So they can survive within the phagocytic cell. Within that phagosome, we have some of these oxygen-dependent compounds. So you have these reactive oxygen species or oxygen-free radicals, these activated halides, nitric oxide. There's also acids, lysozyme, ion chelators, protein that damage the membranes of the bacterial cells. So it's a super, super harsh environment. And so when you have a microorganism that's just like, yeah, this is fine, no problem, it can be a big problem. We can even consider the normal flora as part of our innate immune response. And that's often because it basically trains the immune system, and it helps to maintain a low level of immune activation, because your immune system is always there going, hey, there's always some bacteria here, let's keep on alert. And when the situation changes and there are different bacteria there or more bacteria, that disruption in the normal flora, that can lead to the immune system responding because it thinks there's a problem. So again, with inflammation, for the most part, inflammation is good. It means our immune system is responding. It helps our bodies to heal. But every once in a while, it is not good. And it is not good when we start to see really high quantities of these pro-inflammatory cytokines. And when this happens, we get the phenomenon known as septic shock. So when this happens, especially in response to an infection, that's septic shock. You can have thrombosis, significant decreases in blood pressure, and severe metabolic disturbances, so decreases in blood glucose. This can be fatal. And so that's why we want to monitor an inflammatory response. So I have 2 small children. We all got COVID in December, and we monitor their fever, because if the fever goes, above 102, 103 for an infant or toddler, that's too high. That can lead to septic shock. For an adult, we're looking 105, 106, leading to septic shock. So that's why we monitor those things, because sometimes our immune system is trying to do too much, and we want it to tone down just a little bit. Again, here are some of the mediators that cause the different symptoms we see or the different phenomena we see during an inflammatory response. So the increased vascular permeability, vasodilation, pain, tissue damage, fever. And then in chronic inflammatory responses, that's when you're also going to start to see cells of the adaptive immune response come in. I'm going to not necessarily read this out loud to you. Some of them I will, but there are people who have immune systems that just aren't responding properly or the way that we would want them to. And they can have chronic inflammatory conditions. And people can have chronic inflammatory conditions all over the body. This particular slide is about in the skin. So things like acne, atopic dermatitis, and eczema can be because of inappropriate immune responses or overreactive immune responses. So you can read this at your convenience and kind of see how some of these conditions can arise when things are out of balance. So in general, we have our acute phase response. This is like right when we get an infection or an injury. That's when you get those host defenses that can include things like fever. And as we saw in previous slides, we have numerous proteins that can mediate that response. What's scary is when we run into something like sepsis and then a cytokine storm. So that's when we have the overwhelming release of cytokines in response to infection. And sometimes this can be simply because the immune system is overreacting. Sometimes it can be because the infectious agent can produce molecules that lead to the overreaction. So we'll talk about superantigens and how that can cause an overwhelming immune response. So here they're just showing different molecules that can lead to various effects. Most often, with these kind of thick, big arrows, we can see how strong the response is. Our body, just in general, tends to respond more negatively to gram-negative bacteria, and specifically the LPS. And that's why we call it endotoxin, because it's a toxic molecule for our bodies. So it leads to a really strong immune response, high levels of complement. And so most often, now we're not talking about toxin production or anything, we're talking only about just the bacterial infection itself. We see this response with gram-negative bacteria that can lead to vascular leakage, fever, neutrophil activation, a systemic inflammatory response syndrome, heart failure, and even death. Gram-positives can cause it too, generally when they produce a specific toxin. But this is what's happening when people have a cytokine storm, when their immune system has overreacted to what is found in the body. And finally, because this is the innate, I hope you've imagined that the antigen-specific is coming next. And so that bridge that we talked about before is really the dendritic cell. And the dendritic cell is going to sample antigens and can lead to the direct activation of cells of the antigen-specific or adaptive immune response.